Okay, I'm going to go ahead and get started. Well, once more, welcome everyone. This is the Research Roundtable, Energy, Transportation, and Habitat Nexus. My name is Prarie, and I'm a graduate intern at the University of Illinois, Chicago. And I'm very excited to welcome you all today to the kickoff of this year's Research Roundtable series. The slides. Great. We are very excited to kick off this year's Research Roundtable webinar with a topic that's gained traction a lot over the past few years. And this topic is advanced monitoring techniques for habitat management and right-of-way. And we've been thinking about this topic and thinking about the range of research that's being done. And we're very excited to have a number of presenters here to provide some insights into some of the advanced monitoring work that they are doing. And we look forward to having some interesting discussions with you all on this topic. It takes a while for the slides to get moving. Our objective with this roundtable is to, one, through the presentations we will hear today, highlight current research related to advanced monitoring techniques for habitat management and right-of-way, facilitate discussion about other current and future research, and then create an opportunity to identify research needs and spark conversations that may lead to collaborative work. Oh, too fast. Today's hosts are Ashley Bennett with the Electric Power Research Institute and Toby Chu with Southern Company. Just a few housekeeping items. Please keep your mics muted and video off, except during the breakout session, where we encourage you to turn your video on if possible. If you can, could you please update your Zoom name to include your organization? And you can do this by hovering over your name and clicking the 3 dots on the top right corner. If you're having any technical difficulties, please feel free to reach out to me via the chat box. For any questions or comments that you might have for our speakers, we will have a Q&A session following the 3 research presentations. We are recording today's session and we will share it afterwards. For today's agenda, in the first half, we will hear from our 3 presenters, and this will be followed by a Q&A session. For the second half, we will break out into small groups where we have some great questions to continue the conversation on advanced monitoring techniques for habitat management and right-of-way. And we will reveal the conversations in the breakout session in a very large group recap at the end. And now I will pass it on to our co-host Toby Chu to introduce today's speakers.
Thank you very much. I'd like to welcome and introduce Dr. Ashley Bennett. She's the research lead from the Electric Power Research Institute. Dr. Mark Davis, he's a conservation biologist at the University of Illinois at Urbana, Champaign. And then Erica Christiansen, last but not least, she's an associate wildlife biologist at Stantec. So first off, Dr. Ashley Bennett is going to speak about plants, pollinators, and birds, an overview of monitoring technologies for rights-of-ways. You may be on mute, Ashley.
Can you guys hear me now?
Yes.
Okay, and I seem to have lost my camera for, let's see. There we go. All right, so thanks everyone. So today I'm going to be giving an overview of monitoring technologies, of which I'm an expert in none of these technologies, but I have the privilege of working with a lot of different technologies in my role at Prarie. So I'm going to go through these different monitoring technologies pretty quickly today. So hold on. And I see there's a delay here. So when we're thinking about monitoring technologies, we can start from the simplest, which is just going out and doing visual observations, to passive sampling techniques like camera trapping, to acoustic monitoring, moving to eDNA, to remote sensed data that we can collect through satellites, manned aircraft, or unmanned aircraft, UAVs, or drones. Today, I'm going to be focusing on 3 of those technologies: remotely sensed data, acoustic monitoring, and eDNA, and hopefully giving some practical examples about how each of these technologies can be applied on rights of way. So today, I want to start with remotely sensed applications focused on satellites as well as unmanned aircraft. So when we're thinking about satellite imagery, usually this is going to be lower spatial resolution data collected at higher altitudes, typically at the meter level. We're going to be able to cover much larger geographic extents than we can with drone imagery, ranging from regional to global. We're able to get very consistent data collections, which is determined by the orbit of the satellite, but we do need to have cloud-free days for really good satellite imagery. So the trade-off with satellite imagery is we can get much larger geographic coverage, but at lower spatial resolutions. We have a range in cost from open source data that's freely available to commercial satellites, which you have to pay for that imagery. Now, in contrast with our drone imagery, we can get much higher resolution because we can fly at lower altitudes and lower speeds, resulting in very high level resolution centimeter level data. The geographic extent we're going to be able to cover is much smaller with our drone imagery, typically because we're limited by battery life. We can go out and do collections whenever we want with drone imagery. The trade-off being we're getting much higher spatial resolution data, but covering a much smaller scale. And again, the cost is going to depend on the type of drone you use and the type of sensor you collect. So when we're thinking about satellite imagery and drone imagery, we're going to have differences in spatial resolution, temporal resolution, and spectral resolution. And some of the sensors, I'm not going to really talk about manned aircraft today, but some of the sensors I'm going to cover for drones can be applied with manned aircraft. So when we think about our sensor categories, we have 2, passive sensors and active sensors. Passive sensors are basically measuring natural light reflected from an object. And we can collect imagery in the visible, near infrared, and shortwave infrared. And we can get different spectral resolutions, as I mentioned. And this basically means different wavelengths or bands of light are being collected. So some of you may have heard of panchromatic imagery, which is basically our grayscale imagery, or true color imagery, which is imagery collected using red, green, and blue wavelengths of light. We also have a lot of sensors out there that collect multiple wavelengths or bands of light, from multispectral sensors, which are collecting 3 to 10 bands, to hyperspectral sensors, which are collecting over 100 bands or wavelengths. In contrast, we have active sensors, and active sensors are emitting their own energy source and measuring the returning backscatter to the sensor. So a really common example that we see being used on right-of-ways are LiDAR sensors. So LiDAR sensors are sending a pulse of light that, for example, may hit a tree branch, and the sensor is measuring the return of that light pulse, giving us a distance, for example. So LiDAR imagery can give us 3D point clouds to let us look at infrastructure or calculate height. So this is by no means an exhaustive list of sensors that can be used with drones, but these are the sensors that I have personal experience with drones, ranging from multispectral sensors to hyperspectral sensors to different LiDAR sensors. And again, these sensors are collecting wavelengths in the visible and near infrared wavelengths of light. In contrast, this is a table of different satellites. This is, again, not a comprehensive list. I took this list from Rose et al. 2022. If you want to see a more comprehensive list of satellites, but I basically wanted to illustrate with this table different satellites and how they are different in their temporal resolution. So how often they're collecting data. You can see Landsat 8 is collecting imagery every 18 days. Well, we have satellites like WorldView 3 that are basically collecting data daily. You can also see these satellites differ in the different spectrum or wavelengths that they're collecting data and their resolution. So if we look at Landsat 8, for example, we can see that it's collecting blue, green, and red wavelengths of light at 30 meter resolution. In contrast, WorldView 3 is also collecting blue, green, red, and near infrared at 1.24 meters. So how can we use some of this remotely sensed data on right-of-ways? Well, the first and most common way people can use remotely sensed data is to assess at-risk vegetation. So looking at trees, for example, that might be stressed and pose a fallen risk to right-of-ways, either utility infrastructure or roadways. And we can usually thinking about using multispectral data collected either by drones or satellites. LiDAR is also very important here to help us, again, calculate tree height to be able to calculate fallen risk. We can also use multispectral drone and satellite imagery to look at encroachment of vegetation onto right-of-ways, calculate vegetation diversity, so kind of a metric of habitat quality. There's references using hyperspectral data to do this. We can also use true color and thermal imaging to help us identify different species, from birds to plants to bats to tortoises. A lot of us are thinking about using pollinator habitat on right-of-way, and so we're wanting to know, well, how much floral resources do we have on our right-of-way? So when are we getting flowering? How much flowering are we getting? What species do we have out there, in particular milkweed? So again, we can use multispectral, both drone and satellite imagery to help us answer these questions about pollinator habitat, as well as invasive species that may be degrading our pollinator habitat. And lastly, we can use remotely sensed data to help confirm management. So we can use satellite data to see if a mowing has occurred, or drone data to see if an herbicide application was effective, or to confirm whether or not we perhaps have soil erosion or water runoff issues during maintenance activities on our right-of-ways. I don't have time to go over these references, but wanted to include a list of references that highlight how we can potentially use remotely sensed data on right-of-ways. Okay, so now I'm going to transition to acoustic monitoring. So acoustic monitoring is a type of passive monitoring that's helping to detect and record species vocalizations. Acoustic monitoring can help us identify the presence of different species, identify those species, help us determine the range of where those species are occurring, and their phenology. Typically, we're targeting groups like birds, bats, frogs, and insects with acoustic monitoring. Acoustic monitoring can also help us identify activities, for example, poaching, that by listening for gunshots, for example. Another metric that we can use for acoustic monitoring is soundscapes. Soundscapes are where we track all of the biological, geophysical, and anthropogenic sounds in the environment. And we're trying to characterize biodiversity over both space and time. So we can calculate acoustic indices similar to species biodiversity using acoustic monitors that help us determine change in species over time, as well as responses of species to management practices. So what are some of the advantages of acoustic monitoring? Well, acoustic devices can help us detect multiple species using the same device. We can collect data both during the day as well as night, which can be very beneficial for many nocturnal species that are hard to detect and record. We can detect over both seasons as well as across seasons. Acoustic monitors are easy to scale up, so scaling over large spatial scales. We can sample in difficult to access areas, as well as areas that are impacted by disturbance. Acoustic monitors are also very easy to couple with other technologies like camera traps, as well as drones. So what are some of the challenges with acoustic monitors? Well, first, not all species produce sound that we can record with acoustic devices. Detection can be limited by both the propagation of sound through the environment, as well as any interference we might be having in the environment. Determining true species is difficult. So is that species actually present? Was it sound masked? Was it too quiet for us to detect? Or were we simply sampling in the wrong season to be able to detect that species? Estimating abundance is difficult with acoustic monitors. And okay, I've got a 5-minute warning. Thank you, Toby. We can also have big data issues with acoustic monitoring, from storage processing, and archiving this data. We still have to have field visits with acoustic monitoring, and analyzing this data can still be difficult in terms of extracting relevant sounds. So here's an example of a sampling for bats using both drones as well as thermal cameras, acoustic monitors, and microphones. And since I'm running a little short on time, I'm not going to go over this example, but if you guys have questions about it, please ask me at the end of this presentation during the Q&A session. So again, here are some examples of acoustic monitors for both bats on right-of-ways as well as birds. Okay, so really quickly, the last type of monitoring I want to touch on is environmental eDNA. eDNA is DNA that's shed from a host organism into the environment. We have kind of three different groups of eDNA: botanical, coming from plants, soil eDNA, and airborne eDNA. So we can sample for eDNA from the soil, the air, and water. We can also sample organisms themselves, thinking about insects like this bee and taking pollen off of its leg. We can also look for skin, hair, sample feces, honey, resin, flowers, and flower plant parts to be able to detect eDNA. So eDNA processing has two main approaches: barcoding and metabarcoding. Barcoding uses species-specific primers to detect DNA fragments. Conventional PCR is used to detect species presence, while quantitative PCR is used to quantify species abundance, and it's very effective in detecting invasive, rare, and cryptic species. Metabarcoding, in contrast, uses universal primers to detect millions of DNA fragments. And this approach is very effective in characterizing both past and present biodiversity, trophic interactions, dietary preferences, and ecosystem health. So some of the advantages of eDNA are it's generally less time and labor intensive compared to more traditional field approaches. It's less destructive. So think about sampling for pollinators. We don't actually have to collect and kill the bee in order to identify it. There's no observer bias. Again, when we're out in the field doing pollinator observations, we don't need a taxonomic expert for identification. We can potentially document more species, especially rare and cryptic species. We're able to detect invasive species early, which is particularly important for IVM practices, and we can do concurrent sampling. So what are some of the disadvantages? Well, presence and absence can be misleading. We can get a DNA hit, but other more traditional methods may not actually be detecting that species. We can have bias in detection due to incomplete reference databases. We can get false positives, and Mark can correct me, but my understanding is there's no kind of direct link between our eDNA and being able to understand abundances and biomass out in the landscape and the environment. And we still have a limited understanding of eDNA in terms of persistence in the environment, rates of degradation, and transport. And this is a growing field where we're still developing field methods as well as laboratory methods to be able to detect those species in the field, as well as detect them through bioassays in the lab. So what are some of the practical applications on right-of-ways for eDNA? Well, probably one of the most exciting for me is being able to do rapid biodiversity monitoring. So we're currently doing a project at Prarie looking at traditional sampling to eDNA on rights-of-ways for pollinators. You can also think about sampling bat roosts and what kind of bats are using artificial or tree roosts. Thinking about nesting sites, for example, the gopher tortoise or desert tortoise, and what other species are using those burrows. Being able to detect rare or invasive species with eDNA, looking at the impact of construction or maybe herbicide use on soil biota. One of the most exciting ones for me is thinking about how we can refine our seed mixes for right-of-ways and for areas of the country where we maybe don't have a good understanding of native plant pollinator interactions and what plants are favored by some pollinators. You know, can we use eDNA to help us understand what flowers and plants we should include in those seed mixes? So here is, again, another list of references. Two of these references are by our next speaker, Mark Davis. So Mark, I'm going to pass the ball over to you and let you tell us a little bit more about pollinator eDNA, and certainly correct anything I got wrong explaining eDNA. So thank you.
That's a good segue. So I'd like to introduce Dr. Mark Davis. He is going to talk about
eDNA a little bit more as a tool for rapid assessment of pollinator communities.
Thanks, Dr. Bennett. That was a great primer. So now we'll get really into the weeds on eDNA as a tool for monitoring. There we go. So slides are working. Something, as was just pointed out in the last slide, that my lab group has been thinking about a great deal, and a number of reasons for this. We're recognizing that there's a global arthropod population decline, with pollinators being hit particularly hard. We've seen the news story in the New York Times about the insect apocalypse. We recognize that in terms of the endangered species list here in the United States, that pollinators are perhaps among the fastest growing group of threatened and endangered species, affording federal protection, which of course has requirements for monitoring and for mitigation. But we recognize the value of these organisms providing critical ecosystem services, approximately 24 billion annually to the U.S. economy, and this has downstream implications. And so we're recognizing that fine-tuned monitoring is increasingly important in the Anthropocene, wherein we have increased consideration for threatened and endangered pollinator species. And we recognize that conventional monitoring is effective. However, it is also time-consuming and expensive. As Dr. Bennett pointed out, it's also exacerbated the taxonomic impediment. Simply put, the taxonomic expertise required to identify specimens in hand is increasingly dwindling. We're lacking that expertise. What is out there is few and far between and increasingly pricey. And so can we be innovative? Can we be creative? Can we be entrepreneurial, leveraging novel technologies that are emerging to try and fill these gaps? And, we do recognize that in certain contexts, conventional surveys can be ineffective. I noted in Dr. Bennett's slides, and something that I often think about is, there's a number of shortcomings of eDNA that were listed. But I think it's important to highlight that conventional methods also have shortcomings. They are imperfect in terms of detection probabilities and other things, and you can see them here. They're often time, labor intensive, expensive, with low detection rates. They can be invasive, they can be destructive, they require taxonomic expertise, they can lack flexibility and have other specific implications. So they're imperfect too. And so by building out our toolbox, we can add value in a number of ways. And I hope to illustrate this today with environmental DNA. Again, this is DNA shed in the environment, which can be sampled and analyzed to detect species without requiring efforts to observe, capture, handle, or identify them at the time of survey. Simply put, and again, thanks to Dr. Bennett for priming the pump on this. There's two different pathways that you can use. You can use single species qPCR assays, which tend to be a little bit more sensitive, or you can use metabarcoding, where we're really interested in uncovering the broad community that may be in place at a given sample and location. And it looks like on the bottom image, you go out, you collect bulk samples, be it water, DNA from flowers, leaf litter. You bring it back in the lab, or in some cases, your hotel room to filter out the DNA. And then we bring it to the lab, and we're able to extract and harness that information using these two pathways up above to get a really fine-grained perspective sometimes of the potential communities that are at play. So our work here started off with the simple question, can environmental DNA provide a faster, more cost-effective means of rapidly documenting pollinator communities, particularly in remote locations? The genesis of this work was really with the Department of Defense that is responsible for stewarding their natural resources, but range closure times can be a problem. And so thinking about ways to speed up that process while still capturing that fine-grained perspective of biodiversity. We're still partnering with the DoD on this, but also Department of Energy and the Department of Interior. And so a lot of different contexts within which this technology might be viable. And so our first real foray into this was really trying to understand the efficacy of this. Is it something that could work? And we wanted to try this in controlled settings, but also in the field. And so we established a mixed flowering plant community in a greenhouse. We introduced bumblebees, and then we tested to see if we could capture their DNA. We also validated this in field, comparing with conventional approaches. So I just mentioned we did this phase flower introduction. We sampled using a number of different methodologies and technologies to try and pin down, number one, if this works, but number two, what might the best practices be to maximize our chances of success in using these methodologies and technologies. And we were successful. Good news. And again, Dr. Bennett cited our 2 manuscripts where we detail these things. Indeed, we released common eastern bumblebees into the greenhouse, and of course, we were able to detect them. Great news, the technology works. In addition, we were happy to find that we detected other arthropods that were known to occur in the greenhouse or suspected to occur in the greenhouse, but really weren't formally documented. And you can see a number of those here, including western flower thrips. Keep that in mind, that'll come up again later. Silverleaf whiteflies, cotton aphids, yellow garden spiders, a number of other species that occurred in the greenhouse. And so in addition to the species that were known to occur, we documented additional biodiversity that I will note follow-up visits and conventional sampling has confirmed their presence in the greenhouse. So great, as a proof of concept, we recognize that this technology indeed works. But the field is an entirely different beast. And as Dr. Bennett pointed out, there are a number of biotic and abiotic factors that can wreak havoc with how well this technology and these methodologies work. And so for the last couple of years, we spent the summers up at Fort McCoy in Wisconsin, documenting pollinator communities. And so what we wanted to do is start doing these direct comparisons with more conventional methods. And so in this case, we compared eDNA metabarcoding with camera trapping. And so we deployed camera traps out on focal flower species, and we collected data from those for a period of 3 to 5 days. At the end of that period, we then sampled the flowers, so sampling those same flowers that were camera trapped using environmental DNA techniques. And so to do this, or this facilitated both a comparison between camera trapping and eDNA, but also allowed us to start exploring interactions of plants, pollinators, and indeed other taxa. So we found some interesting things. Camera traps and eDNA give us somewhat different results. Cameras, not surprisingly, were really good at capturing those very showy, large lepidopterans and large beetles and other things. And so big, ostensibly charismatic arthropods were detected readily with camera traps. eDNA got some of those, but it also captured some unseen biodiversity, at least with respect to camera trapping. So again, thrips emerge and a number of other smaller type species that weren't picked up by the camera traps emerged with eDNA. And indeed, they give us pretty different communities. So overall, eDNA detected 55 species, whereas camera trapping detected 22 species, and only 4 overlap between the two. So in general, it seemed that camera traps favored those larger, more conspicuous, often lepidopteran species, where eDNA favored species that seem to have longer interactions or handling time with those flowers. Now, we also recognize that there remain some challenges that, again, Dr. Bennett did a great job of pointing out. There are primer biases and eDNA biases that can impact downstream results. It seems like our primer sets are less effective at detecting Bomba species and some groups of lepidopterans, and we're refining new primer sets to be able to document that biodiversity. But it's imperfect, and it's something to be mindful of. Again, we're not proposing this as a replacement to conventional means, but another tool in our biologist's toolbox. At the end of the day, our results were able to reveal pollinator communities in both controlled and uncontrolled conditions. Both observed and unobserved species were effectively detected, validating the potential of the approach and recognizing that, eDNA captured a broader, although different constellation of taxa. We've developed a comprehensive workflow that we continue to refine, but we also recognize that many factors, biotic, abiotic, and technical, can influence those pollinator communities that are revealed. I think there's a number of benefits to this methodology and technology. Certainly speed is important. It reduces the duration and number of site visits. We can simply cover a lot more ground with a lot fewer people using eDNA. Decreased reliance on taxonomic expertise and more reliance on bioinformatics provides some technical advances. However, that forms how we need to be thinking about training the next generation of biologists, where we focus in on these bioinformatics skill sets so that they're able to analyze these large sets of data that are generated. We recognize that there are some cost efficiencies here. Reduced field time generally confers savings. And also, again, as Dr. Bennett pointed out, there's flexibility. You can begin to merge both the eDNA from the flowers and pollen from the pollinators themselves to begin to understand these complex networks and interactions that are at play within these systems. So we also believe that this is broadly applicable across systems, both as a preliminary screening for rare, threatened, and endangered species. So you can use this as a way to strategically target more conventional surveys where you have some prima facie evidence that a species that is at risk might occur. And we also recognize there are opportunities beyond arthropods. I'm excited that in a couple of weeks, I leave for Hawaii for 2 weeks to deploy this for birds and bees, followed by bats in Arizona. And so this transcends taxonomic groups, which each require often their own unique and technically challenging types of conventional monitoring. And so there are a lot of opportunities to expand this beyond simply arthropods. And again, it allows us to begin to see some of those interactions. I didn't go into it in great depth, but certainly with our work in Fort McCoy, we are uncovering a lot of different interactions. Pollinators, their predators, parasitoids, etc. And so beginning from a single environmental sample to understand these complex interactions that are occurring on a single plant. If you're interested, again, thanks, Dr. Bennett, for pointing these manuscripts out that we've published. You can track these down. I have a number of people to thank, but I saw that my time is about up, and so I look forward to the Q&A in a few minutes. And I don't know how to give up control of my screen.
Thank you very much. That was very interesting. Yes, if people have questions,
You can put some in the chat, or we're gonna just wait at the end of Erica's presentation. You can ask them as well. So let's go again, last but not least, with Erica Christiansen. She's gonna talk about remote sensing, including a pilot study for quantifying nectar resources. This is along Florida's transportation right-of-way. So thank you.
Thank you for the introduction. All right, good afternoon, everyone. I'd like to first start off thanking UIC for the opportunity to present today. My name is Erica Christiansen, and I'm a biologist with Stantec Consulting Services based in Madison, Wisconsin. Today, I'm going to take us from Dr. Davis's ground sampling into the sky, or in space, rather. I'll be sharing the findings of the first year of remote sensing pilot study we've done with the Florida Department of Transportation using satellite imagery. This pilot project aimed to identify where flowering nectar resources occurred along select transportation corridors in North Florida using satellite imagery. The purpose of this project was to assist the Florida Department of Transportation in identifying monarch butterfly conservation areas along their system to support the nationwide candidate conservation agreement with assurances for monarch butterfly on energy and transportation lands. Our primary objectives for this project were to, 1, identify locations of blooming wildflowers, 2, to quantify the density of these flower patches using percent cover. And we also wanted to understand the strengths and limitations of this type of remote sensing compared to traditional field methods. Stantec worked with a satellite company to procure 50 centimeter resolution satellite imagery of the desired locations, as well as time and environmental conditions. As Dr. Bennett alluded to earlier in her presentation, satellite imagery has a number of scenarios that either make it more successful or not in how that imagery turns out for analysis. So we specified the conditions that we were looking for to best optimize the capture of wildflowers in our imagery. So some of those considerations includes cloud-free timing, time of day when the blooms would be most open and visible, and the bloom period in spring in North Florida, which is late April to May generally. We provided a shapefile of selected roadways with a 500-meter wide buffer. These roadways had been selected in February of 2022 based on coordination with FDOT's central office and district maintenance offices who are familiar with the historical vegetation conditions of the region. The scope of the imagery capture was to be taken during the optimal blooming period for the region to best capture those times. Once we received the imagery from the satellite company, we stitched together the images to create a seamless corridor and reduced the 500-meter wide buffer down to the right-of-way limits. I'll add that this clipping down to the right-of-way wasn't a perfect science since DOT doesn't have the legal limits of their right-of-way digitized across their entire system, but we use some informed decision-making to limit down that imagery so that we're only really analyzing the imagery that's relevant to FDOT and not what's beyond the right-of-way. Once we had our imagery corridor, we began our object-based image classification to identify and quantify wildflowers. This classification primarily used spectral and textural values to distinguish wildflowers from other vegetation or infrastructure within the right-of-way. So we used RGB and near-infrared imagery to make those classification models. We developed a classification model to run across the corridor that was trained using a combination of field collected data. So similar to Dr. Davis's presentation, we didn't do this pilot study in a silo. We also went out and did some ground truthing to be able to compare our methods to more traditional methods, as well as using some mobile mapping data and historical images. So our results were very encouraging for the corridors that we analyzed. Our classification model analyzed just shy of 4,200 acres. Of those corridors, we found that 51% of them were vegetated. So meaning the road surface or the hard pavement shoulder took up 49% of those acres that we analyzed. Of the vegetated acres, we found that wildflowers made up 25 to 35% of that vegetation at varying densities. So you can see in this graph here, the dark blue is the areas of dense wildflowers, which we considered areas, wildflower patches that were greater than 25% or more percent cover. The orange is sparse wildflowers where there was less than 25% cover, and then non-flowering vegetation, which is primarily grass in this instance, made up the bulk of those proportions. So just going through some example areas here along I-10 and the Panhandle, you can see this slight pink shading, which if you zoom in, you can see how the object-based image classification categorized these patches of wildflowers. So in this image at this interchange with I-10, you can see dense wildflowers shown in the dark purple, which was greater than 25% cover, versus the light purple was less than 25% cover. And then the empty spaces were where there is no wildflowers detected in those areas. Similarly, along I-75, and then a smaller US highway. Now, I mentioned that we did some ground truthing of these images. One way that we did that was using a vehicle mounted camera to drive some of the corridors that the satellite imagery was taken on. So the image on the left shows what the satellite imagery looks like, the photography by itself. So from the naked eye, you can detect these patches of dense wildflowers, and we were able to ground truth. And you can see these same patches show up in the model classification. Versus our mobile mapping data on the bottom right, you can see where these patches show up, but you can also see where the model was more successful in detecting these really sparse patches across the right-of-way corridor. Vice versa, here's an area along the imagery corridor that did not have strong detection of wildflowers, and we could cross-check that with the bottom left, showing indeed we really did not see wildflowers along that stretch of roadway. As I alluded to earlier, we also completed some field sampling in conjunction with this remote sensing study. We sampled 60 random locations along North Florida during the same time period as the satellite imagery collection. Some sampling locations did fall within the imagery corridor, but overall, we're located more broadly across the 2 FDOT districts to get a broader basis of what vegetation conditions were like across the region. This field collected data set offered a perspective to the strengths and limitations of our remote sensing study. The key takeaway was the amount of data that we collected. So our 60 field sampling locations, each sample was 1,500 square feet, and we sampled using the tier 2 version of the pollinator scorecard. Even with 60 sample points of 1,500 square feet each, this only adds up to about 2 acres of roadside sampled, whereas our remote sensing study captured and analyzed over 4,200 acres. Remote sensing also provides some answers to questions presented by field collected data. For example, how large of an area is the location of this habitat sampled? So beyond just the boundaries of our sampling location, how far does that extend across the corridor? How uniformly distributed is this habitat? And on the other end, when we don't find habitat, why did we not find habitat in this location? Where's the nearest location of habitat relative to the sampling location? Is it because it was just mowed? Is it because the habitat is located on the opposite side of the roadway where the sample did not fall? Those types of questions can be answered through use of remote sensing. So in summary, again, our field sampling really only sampled 2 acres across this really broad region of North Florida. And the results were about middle of the road, where 55% of plots had greater than 10% cover. Whereas our remote sensing was able to analyze just shy of 4,200 acres. 9% of that vegetation had dense cover of nectar plants, and 22% of vegetation had sparse cover of nectar plants. So some lessons learned from this pilot study. Similar to eDNA, it can be a more cost-effective tool and time savings. You also get much more data. In the context of sampling along transportation corridors, it's also much safer to do this remote sensing rather than sending people to be alongside a high-speed roadway. Some limitations of satellite imagery and object-based image analysis. Canopy cover was one of the biggest limitations that we found, particularly in Florida, where there is a lot of forest cover. Either tree limbs or shadows from trees can obscure the ability to analyze wildflowers beneath those areas. And so we took that as a lesson learned for our improvements for next time. The strength and accuracy of our classification model was highly dependent on the conditions for when the imagery was taken and the availability of ground truth data. So we're doing a second year of this pilot study this year to incorporate some of these lessons learned. First, we did some GIS pre-work to identify more favorable corridors that seem more conducive towards satellite imagery by narrowing our areas to locations with limited adjacent forest cover along the right-of-way to eliminate possible shrouding of the right-of-way from shadows or tree limbs. Second, we created a more adaptable approach of ordering and collecting imagery by first communicating with local staff to report out on current vegetation conditions, for example, blooming rates, mowing schedules, and then strategizing the timing of our imagery order. And third, we're collecting as much ground truth data as we can along these corridors using mobile mapping data collection through vehicle mounted cameras. We're in the process of collecting our second year of data this month, so we'll be hopefully able to share our findings at the end of the year to see how successful these efforts were. A quick thank you to our team that's worked on this, both at the Florida Department of Transportation and at Stantec. And now we'll be wrapping up the presentation portion and I believe going into our Q&A session.
Yes, thank you so much. And hopefully we can have you back to present those second year results. If y'all have any questions, y'all can speak up, go on, unmute yourself, or put your questions in the chat. While I wait, I will ask this starter question for our speakers. It says, what do you see as one of the biggest or exciting opportunities for applying these technologies and research to the right-of-ways in the next 3 to 5 years? What do you envision? Like, how is this tech going to get better, and what can we use it for?
So I guess this is Ashley. I'll take a stab at that from just an overarching perspective, thinking about both remotely sensed data as well as eDNA. And I kind of touched on it in my talk, but it's kind of this rapid monitoring of biodiversity or habitat. So I see both remotely sensed data, in particular satellites, as Erica just talked about, helping us assess habitat quality and flowering resources. I'm always thinking about pollinators, so pollinator habitat, but then what Mark talked about, again, being able to use eDNA to do these kind of rapid assessments of what kind of bees and butterflies do we have out on the right of way for both the remotely sensed technology as well as the eDNA. I think those are both super exciting areas of research, and the technology, both our sensors are going to keep getting better and higher resolution, and our ability to detect those species and those assays and improving those field sampling techniques that Mark talked about are only going to continue to get better. So I see a lot of opportunity in both of these spaces, and I'll let Erica and Mark chime in on their thoughts.
Yeah, I'll jump in and kind of take the next step. For me, I think the really exciting aspect of the eDNA piece is that, we're moving well past this proof of concept. Does it work? And now it's to deploying for comprehensive analyses. And so we can expand our thinking beyond, narrow categories of taxonomic groups, right? And so deploying multiple types of sampling, airborne eDNA is a big one, along with soil, with water, with other things at a single site, being able to survey for plants, for arthropods, for vertebrates, the whole shebang-a-bang. We're going to be able to do this, and we are doing it. And so, we can move beyond this intensive focus on perhaps single species and begin to get a more compelling perspective of broad communities, number one. And number two, we can look at management practices and how those communities are changing relative to those management practices. We often think that, if you build it, it will come, right? If we do these practices, these things are going to come in. But we can begin to look at not just our target taxa, but the broader communities using varied approaches of eDNA sampling to get that perspective. And I think that's really exciting.
And I'll just conclude with a couple points specific to satellite imagery. I think, as Ashley alluded to, sensors are improving year by year. Imagery is also getting cheaper as those improvements happen. And as more satellites are sent into space and privatized, there's more options to collect imagery at a more frequent basis, just allowing this process to get easier and easier as time goes on.
Okay, thank you so much. I see a hand raise. Leonard, if you'd like to unmute yourself and ask your question.
Sure. I'm curious, particularly for Erica. I was curious if the the NDVI from your remote sensing generally correlated with more biodiverse plant populations or more wildflower coverage? In other words, are the healthier, more vegetated areas, generally speaking, also generally more biodiverse? Maybe that's an oversimplification, but was curious if you noticed any kind of quantitative correlation there.
Yeah,
that makes sense. Hopefully that question makes sense.
Yeah, I think I follow. That's a great question, and I'm not sure our study was designed in a way to necessarily answer that. One of the limitations of satellite imagery remote sensing is the ability to detect on a fine scale distinctions of, species diversity in plants. Generally, just through local knowledge of the area and the rights-of-way conditions, we know that most wildflowers along FDOT's right-of-way are either Coreopsis or phlox species. But there are some naturally occurring wildflower areas as well that might have greater biodiversity. Those areas could potentially be more difficult to detect because those wildflowers may be less showy than Coreopsis or phlox. So I think that's a question that I'd be interested in investigating further.
Awesome. And maybe one quick follow-up question. Did you do any downsampling to see I saw you use 50 centimeter resolution. We sometimes have feeds that aren't that quite that fine or aren't that high resolution. Have you looked at lower resolutions? Do you think 50 is necessary, or any thoughts on that?
We haven't tried other resolutions. Stantec has a contract with a satellite company, so that resolution at that scale was what we've used in other applications of remote sensing. And so I think that's just kind of what we defaulted to. I have seen some publications, particularly out of Europe, that has looked at that question of, what's the cost trade-off of higher resolution versus lower resolution data. And generally when you get to, sub-meter accuracy, or sub-meter resolution, I should say, there isn't a big difference, I would say, in the literature of better results versus for higher centimeter resolution than just at 1 meter resolution.
Okay, thank you.
Yeah.
Great work.
I see Rory asked about how to sample airborne and plant eDNA, and Mark answered that in the chat. So you can read that, Rory, and if you have any more questions, you can put it in the chat or put yourself on, unmute yourself. But then I did have a question from, I think, Gwen, that maybe they put their hand down.
Yeah, Mark answered the question. That was exactly what my question was.
Okay. Well, Mark, do you want to go into more about this? The airborne DNA. Are you getting any good feedback on that? I know.
Oh, great stuff. Yeah. So
thing with for bats. Are you what are you trying to get? And are you trying to get plant eDNA from the airborne, or what's the deal?
listen, our philosophy is we want to get all the things. That's what we're doing, right? Because you can get it all. And so, yes, we're using, depending on the context, passive traps for birds, for arthropods, pollinators in general, or specifically bats in the desert Southwest. We're deploying both passive and active airborne traps wherever we're doing our other forms of eDNA sampling, again, because it gives us a different perspective. So, we're certainly detecting birds with it, which it's been pretty effective. Plants as well, both threatened and endangered and invasive, so a lot of utility there. Mark Johnson, he's a postdoc in our group that was at Texas Tech, but he's really been doing pioneering work. I encourage you to look up Mark's publications because we're taking what he's built out and expanding it in a number of different contexts. But it's all the things, right? And so far, we've even detected, or Mark's even detected, toads, believe it or not. The sky's the limit. If you have an assay that will amplify it and its DNA is out there, you're gonna capture it.
I have a quick question to that.
Go ahead.
So how where can people send these samples? Is there one location? Is it very species specific? Is it like, where can these be sent commercially?
No idea. I know there are some firms out there that are doing some of this stuff. We're doing this on contract for our research program. I don't know how well it's built out for being able to send to a sort of warehouse for this sort of work. I don't know, honestly.
Can we follow up? I don't know, Ashley, if you were going to ask, but for the toads, is it just the skin sloughing off and getting into the trap, or what's going on with that?
I'll preface this by I'm a herpetologist at heart. I'm teaching herpetology here this semester, and so maybe I think about this stuff too much. This was some of Mark's work in West Texas. And listen, West Texas is rough for amphibians. And sure, it could be from cells sloughing off. These things can have high mortality rates in these ephemeral pools as tadpoles. And so dead, desiccated tadpoles being mixed in with the dust in West Texas, decaying carcasses, there's any number of sources for all of the different types of DNA we're collecting in these samples. It could be fecal material. I have toads that like to defecate in my front yard on the driveway. It's fecal material. All these different things are sources of DNA, and you get strong winds, which you certainly have in West Texas, and it's going to blow it right into your traps, and you can detect them.
Okay, thank you. Anybody else? Doesn't have to be about toads. It can be about something else. Yes, I see Nicole has her hand up.
My question is specifically about the eDNA sampling. So with conventional surveys, there's definitely an education requirement before people go out into the field. So how do you think it compares for something like eDNA sampling, the education requirements for conventional surveys versus sampling this way? Do you think it's more intensive? Do you think it's going to be like if you were to do like a cost benefit analysis almost? How do you think the two compare with each other?
That's a great question. I think eDNA in a certain respect is easier, simply because depending on what you're working on, what your taxon of interest is, things can be very species specific. So I think about it in the context of I do a lot of work down at Fort Polk in West Louisiana for Louisiana pine snakes and alligator snapping turtles. And the ways of sampling for these two things are vastly different, very specific, require specific equipment, and it's really difficult to establish a community science program and do that training. And then it's compounded with these are threatened and endangered species. You can't have someone just walk out into the landscape and start surveying these things and picking them up. They likely won't be able to get those permits. We think of the same thing with our requirements for bat handling here in the Midwest, where we have a number of endangered species. You can't just engage a community scientist group to go out. Now, you can use, similar to eDNA, an acoustic program, which is going to be much more broadly deployable at scale. eDNA is relatively straightforward, irrespective of what sorts of organisms you're trying to sample for. And indeed, we've got a community science project here with the Field Museum up in Chicago, where we're looking at pollinator communities at sort of the urban, suburban, exurban ag interface. And it's great. We can do a one-day training session. We can put videos up of the SOP, and we can have people go out in the field and do those collections and send it to us in the mail. The samples are fairly stable. And so I think that technology transfer piece is much more straightforward. And it gets us around those strictures that are placed on us by the Endangered Species Act, where we can't have people going out to try and handle and capture rusty patch bumblebees. It's not going to work. And so I think there are a number of distinct advantages here.
Thank you.
Mark, how are you? Sorry, I have a question. Mark, how are you having your citizen scientists collect those samples and send them back? Are they collecting flowers and?
Sometimes, yeah. It really kind of depends on the project. We've moved away a bit from actually sampling the flowers, because you do get some PCR inhibitors to leach out of the plants themselves if they're in the buffer for long term. But it's, sending us the sort of wash of the flowers. So putting the flowers in a wash, shaking it, and then sending us that wash, and then we do the filtering and processing. Others have, sent out sort of community science filtering kits where they can filter on site with hand pumps or other things, and then just send the filter in desiccant, and that works as well. For our specific program up in Chicago, they're actually partnering with folks from the Field Museum who are going out and deploying the materials that they need, and then they get the water back, the Field Museum filters them, and then sends us the filters. Helps to have partners with cachet in the community.
Yeah.
I see Leonard has another question. Go ahead.
Yeah, and this question, the panelists may not like it, but it's I find that there's a difference in mentality and mental model of people that are field practitioners, say working along rights of way, and whereas I think in the research community we love seeing all these different data points. I was just curious with looking at all this rich data, like the remote sensing and eDNA that has geospatial complexity, temporal complexity. Have you guys looked at any kind of dimension reduction where you could provide a simple before and after index? Because I feel like users sometimes they struggle to kind of grasp, all the richness of what's possible and all the benefits of new management practices. So just curious about that. Could anybody get down to something like an 8 or a 9, or, if that makes sense?
Yeah, Leonard, can't speak a ton about it, but we have a paper in review where we've generated an eDNA IBI. So I'm not sure if that's exactly where you're going, but really it compared quite favorably for a fish IBI and was super competitive, and in fact revealed a number of things that the conventionally sampled fish IBI doesn't. So yeah, I think so. I think you can begin to generate those metrics to be able to have a standard comparison across landscapes, across contexts, and hopefully that will be published later this year.
And what does IBI stand for?
Index of biotic integrity. It's a metric often used in stream systems of kind of looking at the different communities of invertebrates, of vertebrates, etc., to be able to compare in a quantitative way across watersheds potential degradation or recovery after management practices, those sorts of things.
Mm-hmm. I have a question, but first, Kelvin asks, where are the Field Museum volunteers sampling?
In neighborhoods in and around Chicago.
Okay, great. To that end about integrity, what about thinking about in each type of technology type, like false positives? I know with eDNA, some companies are a bit worried, how rare these animals are, and then say their DNA shows up. Does that automatically mean, the animal's there? And how would you maybe talk about some of the disadvantages of your sampling technique?
Yeah, I would push back on that and say that criticism belies a misunderstanding of eDNA in these systems. It is not permanent. It degrades. And in fact, we've got papers out that shows it degrades relatively quickly. DNA is really blasted apart by ultraviolet radiation. We sure do have a lot of that out there. I didn't get into it with the pollinator stuff, but rainfall washes away eDNA. We found that after a rainfall event, if we snipped a flower and tried to sample DNA, it wasn't there. And so it is not permanent on the landscape. It is ephemeral. And so, sure, does it mean that this organism was there yesterday? No, but unless you are taking sediment cores from lakes, the likelihood of a detection not representing an organism that was there within the, last several months to a year, I think is folly. And a great example that I would show is our work on the Louisiana pine snake. We did a lot of work down there where we detected telemetered snakes that had been in a sampling location within a fairly narrow period of time. And then we went back and sampled those same places in December. And in December, these snakes are much further underground in pocket gopher burrows. They're not surface active. They're not around. In those same locations where we had telemetered snakes during the active season, we didn't detect a single one. And so, this notion that DNA is going to be persisting on the landscapes for years on end for us to harness, I think is where we're doing a bad job actually talking about fate and transport, because it is not forever.
Mm-hmm. And then, of course, there's QA/QC measures, so you won't have contamination, lab sample contamination. that sort of thing.
Yeah, every step of the way we're doing controls. So there's a field control. We're taking blanks in the field. There's an extraction. There's a filtering control, extraction control, PCR controls across the board. And that allows us to assess our false positive and false negative rates and really have a fine-tuned understanding of how good of a job we're doing.
Okay, and I see another question from Tim in the chat. Says, is there a comparison about the plant sampling locations, i.e. the sampling or taking a swab of the basil leaves versus the stem or the flowers, and there's a potential impact for species detection depending where you're sampling on the plant?
Tim, I'm sure someone's working on it. We've been focusing in my group on the flowers themselves, but I recognize that there is a lot of different groups that are working in this space. I suspect that if it's not in review already, it'll be there soon, but I haven't seen any hard data yet. That doesn't mean it's not out there. I just haven't seen it.
And then Erica, if you wanted to touch on it, are there any, I mean, can you have like a false positive in remote sensing, or how do you deal with, or how you have control?
Yeah, so there definitely is some false positives. I mean, the model can, mistake glare as white flowers, for example. So the way that we kind of do our QA/QC process is we look at a subset of the imagery, build the model based off of that. Hopefully we have some other data sets to compare that imagery to, whether it's field collected data or some mobile mapping data or historical images. One great thing about sampling along transportation corridors is Google Street View is always an option, at least in most places. So we build that model on a subset of data, and then we use that to classify the rest of the corridor, and we do spot checking along the corridor to see how well that model is performing ubiquitously across the corridor. We did find last year that, because of things like cloud cover or time of day restrictions, the satellite imagery company would have collected, maybe half of the imagery of a corridor on one day and then had to go back another day to collect the rest of it. And so we would find differences in the model performances because those imagery data was from two different collection dates. And so you have to rebuild a separate model, we found, for those different data points. So that's just an example.
Thank you. Yeah, is there anybody else who has any questions before we go into the breakout session? Here's your last chance, last call. Okay, well, we can move on to our the next part.
Okay, so I think this is me, Toby, to walk us through the breakout sessions. So we're gonna, for the next kind of second half of our webinar, we're gonna have breakout sessions that are about 30 minutes long. We're not gonna record these sessions, and then we're gonna have a short recap at the end of our breakout. So we're gonna have about 4 breakout groups. Each breakout group has a couple of leaders, a technical leader and an industry lead, that will help facilitate the discussion. Next slide. So for our breakout sessions, we're going to use a platform called Miroboard. So here in a little bit, if it's not there already, you're going to see a link in the chat. And when we get to our breakout sessions, you guys can click the link in your chat, and it's going to open up a new window on your computer that will allow people in the breakout to type notes, to ask questions through kind of making a little post-it in the Miro board. So it's great for people who don't want to take themselves off mute or can't, or it also helps us capture notes in the breakout. So we hope everyone turns their camera on and takes themselves off mute, but if you don't want to do that, you can use kind of a little post-it in the Miro board to make comments or ask questions. Okay. So for the breakout, everyone is going to be automatically pushed into a breakout group or breakout room, And then at the end of the breakout session, everyone will automatically be pushed back to the same session, general session. And at the end of our breakout, we're going to have a quick recap of everyone's discussion in their breakout room. So we're going to ask one of the discussion leaders to be prepared to kind of give a recap of what happened in your breakout room. If you have any issues while you're in your breakout room, you can ask for help by either leaving your room, don't leave the meeting, just leave your room, or you can invite the host into your room to help you troubleshoot. Okay, so we already have some questions to help jumpstart the discussion for each breakout group. And I'm going to go over those with everyone here really quickly. So the first one is, which of the technologies covered today do you currently use and how? And which technology would you like to use in the future? Secondly, which do you see as having the biggest opportunity or barriers in terms of the technologies we cover today? And then lastly, what additional research is needed to support the adoption of these or other modern monitoring technologies on right-of-ways? So other technologies we talked about today, what research is needed, or are there other technologies out there that we didn't talk about that you guys would like to discuss? Anything's fair game. So those are the questions. I think that's my last slide. And if it was, we're pretty soon all going to be pushed to our breakout rooms.
Okay, we're just going to give everyone a few more seconds to hop back onto the main room. I'm going to hand it off to Ashley, who's going to lead us in our breakout session recap.
Okay, let's see, getting myself unmuted. And we have about 15 minutes left. So this is the time where I'm going to ask our breakout leads to give us a recap of what happened in their breakout sessions. So for the people that are going a little later, if we start to get kind of repetitive on the comments, I'll ask you just to highlight kind of what's new and hasn't been said yet in terms of comments in your breakout. So let's start with breakout number 1.
All right, that's me, Ashley.
Great.
So we missed you over there because I thought you were supposed to be there with us, but
I know, me too. It's okay. Okay.
Oh, but anyways, we talked a little bit about what some folks were doing, and primarily a lot of us are doing the more of the visual, the LiDAR, and some are starting to step into looking with the camera, the acoustics, and in the beginnings of eDNA. Had some kind of questions, I guess, looking at like the drone with the acoustic monitoring for bats. How does the drone or the acoustic monitor take out the drone noise, and how well do they fly at night if they're flying within visual line of sight? So but then we started to look at some opportunities, and we saw that with eDNA there's some great opportunities there, especially when we start looking at the ET and R species, in particular maybe even bats. Can we use this without the a lot of false positives and being able to identify some of these species that are out there? The group felt that we still needed boots on the ground We still needed that verification out there that maybe as we move into the future, looking down the line, that they're going to work hand in hand. You're going to have to do boots on the ground to verify stuff, and maybe to get the more accurate, you know, really detailed specs that you really need that you can't quite get with us. You know, and let's see. We also talked about how cost prohibitive some of this is. You know, we know that LiDAR was very expensive when it first came out, but as more companies got into doing LiDAR, it started to drop in cost. So is that going to be the same with a satellite or with the eDNA? Then we started looking at some of the research, and you know, you know, hopefully that, you know, as we start looking at more research out there, we can get, you know, more standards set up for eDNA, you know, more useful methodology. You know, we need to make sure, you know, with that future research, that the right questions are being asked in the bid packages, because most of us as veg managers out here, we don't know. We're not LiDAR experts, we're not satellite experts, and we're certainly not eDNA experts. So we need to know, you know, is there a template or something we can use to make sure we're asking the right questions, you know, when we go out for bid, so that we're not getting information or getting stuff that we don't need that isn't going to help us with our programs. A question got brought up as to why right-of-way stewardship or right-of-way stewards aren't more of the conversation here in showing how we're preserving this biodiverse habitat that's out here on the right-of-ways. And then we said that drones and remote sensing may not replace boots on the ground, you know, but it certainly does have a benefit. And you know, and how can we find that equal or medium ground? The other thing that we did mention too was that we need to drag or bring along the regulators to the conversation, because it's really important here. You know, even early conversations with the regulators as to what do they see that they need for research out here, because we as the industry experts seem to be the drivers out here, and we need to bring them on board as well. You know, and I think they are starting to realize that we are the subject matter experts, you know, in the field, in this realm of maintaining and creating this biodiverse habitat out here, and that we don't run scorched earth programs, you know, and that we're trying to protect and keep this habitat that we've created over time. And so we need to bring them to the table and make sure that you know, that if we're going to go eDNA, that we can utilize this, you know, with Fish and Wildlife Service in parts of the you know, protection of protected species out there, or to show that they're there or not there, you know, so that we can move forward with our work. So and then I'll just touch back with Toby to make sure I didn't miss anything. So,
No, I think you covered it. Thank you.
Thanks.
Good.
Well, thanks, Lou and Toby. That was sounds like you guys had a really great discussion and covered a lot of different topics. So breakout number 2, I think, Mark, you were in breakout number 2.
I was, but I think Luce is going to report out for us.
Okay, great.
Yeah, hi everybody. So we had a really great conversation regarding eDNA, since Mark was leading our conversation, and the different agencies that have used eDNA. And so the same thing was problematic with our group. The cost is prohibitive, and more so developing a pilot and a proof of concept, because this is new technology and it's only come about within the last 12 years. Mark was referring to that. And then also bridging that gap between, you know, our practitioners with our DOTs and our science researchers that complete the analysis, and they do all the different types of technologies that we can actually implement in our DOTs, but that information is not being relayed to our practitioners. And then a really great idea came about where we can start, if we can work with our university programs, sort of allowing these new types of technologies, whether it's eDNA or aerial, or, you know, GIS took a while with developing that GIS programs within our universities. to allow new academic courses at these university levels, you know, because there's a lot of students out there that are still studying the same technologies for doing their field work. There's some other information, but I know we're running out of time here, so I'll just close with that. Thank you.
All right. Thank you, group number 2. Let's see. Breakout number 3 looks like was Rory and Erica. Not sure who's going to give you a recap. Me, it looks like Rory.
Oh, I was going to suggest that Erica.
Okay, Erica, that's okay.
Yep, I can take it. Yeah, so we had some great discussion as well, and very similar topics that all of you talked about as well. I think, you know, one of the barriers is, you know, you collect this data, but how do you transform it in a way that is effectively communicating the points or goals that it's trying to point out? Dennis made a really great point as well that, you know, even though some of these remote sensing technologies can be quicker in the data collection than traditional field methods, the time for the data processing of these very, very large data sets may be prohibitive in your ability to act on that. So you go and collect data for a period of time, you have to take a bunch of time to process it, when it possibly in circumstances been faster to have just done it the old way and been able to act on it and respond in a very efficient manner. And then kind of just concluding that even after you've collected this data, depending on who's collecting the data, implementation may not even occur even after you've created this great report of saying, you know, this is what's out here, or this is what we're finding, how can we act on it? And that might fall on deaf ears.
All right. Do you guys have any?
Just to add
Yeah, go ahead.
to that last part, just kind of the importance of, and I think this was mentioned earlier, the either licensing or regulatory or cost information that would help make the case to change management.
Absolutely. Okay, breakout 3. Is that it? On to breakout 4? Okay, I'm seeing a thumbs up. So Nicole and Tracy, I'll let you guys, one of you, give your report out.
Okay, it seems that a lot of people in our breakout group were interested in implementing and using eDNA. That was kind of the trend that we saw. A lot of remote sensing seems to already be used for more like plant compositions. Someone is already using it for germination success. So it seems a lot of places are already implementing some of these technologies for different things. The biggest opportunities or barriers, we did briefly touch on cost. We touched on education. We also touched on restrictions on UAVs and how that could be restrictive to work being done. And then Ashley actually suggested using like the companies that we work for on right of ways, like utility companies, using their imagery instead of going through the restrictive, you know, UAVs and everything like that. So some of the additional research that our group talked about was, again, cost comparisons. So we talked about like using a drone versus satellite imagery, using field methods versus drone or satellite imagery. We talked about how use of these methods can improve safety in the field, specifically along rights of way. And we talked about how the rapid biodiversity assessments can be used in many different ways. An additional thought or comment that was brought up in our group was a question about using AI and remote sensing. And Ashley kind of touched on that. That is not necessarily being done, but the technology is starting to be explored. So looking into more ways to use that. But yeah, I think a lot of groups touched on the main things that we did, definitely cost. And another big one was institutional acceptance and budgeting for use of these technologies.
All right, you guys, thanks to all the groups for their feedback and report back. So we have just a couple minutes left. So I think I'm going to hand it back over to Tega.
Wonderful. Thank you so much, Ashley. Well, I know we're running short on time here, we're almost at 3 p.m., but I would like to thank you all again for joining us today. Thank you for your feedback and your thoughts, comments, and questions. We do invite you to share your thoughts on today's webinar in our survey, and we will provide a link in the chat. Also, we ask that you keep an eye out for upcoming webinar topics for this year. The next webinar we will host will be focused on invasive species management, and we will also host a series of webinars on public perception, communicating with landowners and educating regulators, and vegetation management and traditional ecological knowledge. So thank you all again for joining us today. We hope you have a great rest of your day.
Thanks to our speakers. Great presentations, Mark and Erica.
Thank you.